Alright, hello everyone. I'm Sarah, and today we're continuing our lesson in, I guess, basic scientific things. Uh, Last time we discussed the basic characteristics of life, which are eight, and we said those are what qualifies something to be alive scientifically. Those included having cells, being able to reproduce, being able to metabolize food, which is like you make you take nutrients from the outside like food and sun, and then you make energy out of them to keep you alive and moving. We also talked about homeostasis, which is maintaining the balance in living things, helping them if they go through something, they can always come back to this balanced state. We talked about heredity, which is they're able to pass on their information to the next generation. Um, we also talked about response to stimuli, um, which is the environment around you affects you as a living thing, and you're able to give and take from that environment. Also, growth and development throughout a living thing's, I guess, life. They go through different phases and they grow and develop. They do not stay the same throughout the whole time. And then we talked about adaptation through evolution, which is throughout generations. These living things also change throughout generations as a way to help them, um, I guess, adapt with the environment that's around them, the changing environment that's around them. And these eight things are what qualifies something to be alive. So we discussed the just the cells, like the basic cells. So the prokaryotic cells, which make up like bacteria, and the eukaryotic cells, which make up, for example, humans. And then we talked about what's in them, the little differences, the big differences, and also the main things that they have in common, like they both have DNA and they both have ribosomes, which make the DNA into proteins, which make everything. Um, and we talked about the major differences and similarities, which prokaryotic cells don't have these little compartments, these little proteins enclosed in here. They just have just their DNA and just ribosomes. Very simple. Eukaryotic cells have these different organelles. They have different roles. But both cells are able to divide and use up energy and keep the organism alive. It's just that they have some very basic differences between them. Um, we also talked about viruses. We talked about the very basic form of a virus, what it has in it, which is just its genome, which is like DNA or RNA. So that's the information that it carries. That's what makes it itself, and then um, is just enclosed in this little capsid that keeps that genome together, and then it has these proteins around it um, that kind of send signals as to the virus being somewhere. So whenever the virus is somewhere, it does send signals around. That's how we detect it. Um, and these also help it enter the cells. So today we're also going to be talking about how it does that. So also today, the main thing that we're discussing is mitosis. So mitosis is the process of cell division. So this is how cells divide. This is how they make more of themselves. So when the cell gets to a certain point, it gets these different signals at like a certain point in its life. It gets these different signals to tell it to start dividing. And that's how we maintain our cells. That's how that's how we maintain our life. So mitosis is kind of very simple for the most part. You first just divide the DNA, which is this over here, the information, and then you divide the cell itself, which is right here. You end up with this. You end up with two cells that look exactly alike. So you start with one cell over here, and then you end up with 2 cells. So this is how they divide. So for example, if you look at skin cells in humans, those are always dividing because our skin is the very outside and it's always shedding, it's always changing, so it's always dividing. If you look at other types of cells, like for example, muscle cells don't divide, they just grow in size, but they don't really divide. That's just how they are. So different types of cells divide differently. Some of them don't even divide, it's just the way they are. So let's start with mitosis. So the first phase of mitosis is called interphase. So that's when the nucleolus and the nuclear envelope become distinct, and they form these shapes called chromosomes. So the DNA is first in these little like thread-like shapes, very, very thin threads. These are called chromatin, so that's the shape that they are in. But when the DNA is going to divide, this is kind of very messy. So it's not easy to divide. If you want to divide the DNA, it's not easy to divide this way. It's kind of like messy, and it's not a very good way to divide. So what the DNA does is it condenses into these thick it's the same thing. It just condenses, it becomes thicker and just becomes more compact and more organized, and this is what we call chromosomes. It's in this form of this little X shape over here. So the very first sign that a cell is developing is when the DNA becomes this thick shape called chromosomes. So when it becomes in that form, that's how you know the cell's about to divide. Otherwise, it's usually just in this thread shape, this kind of messy shape. So you know it's going to do that when it becomes in this form, and the nuclear envelope around it, so that's the thing that kept it together, it kind of starts dissolving. Right here, you see it slowly dissolving, so there's a difference from here to here. And then you have these little two things, these little two proteins. These are called centrioles. So these are going to be moving around the DNA for it to divide. These are very, very important. So these have these threads in it. These will attach to the DNA. So after prophase, so interphase, this is how the cell normally looks like when it's not dividing, right? So the DNA is in this form, threads, and then you have a nuclear envelope around it. So prophase is when it starts to prepare for dividing. So the chromatin becomes condensed into these chromosomes, and then these centrioles over here, they produce these threads over here. This is what helps attach, and then the envelope that surrounds the DNA kind of disappears bit by bit. So then these chromosomes over here, they line up right in the middle of the cell, and they line up like that, exactly. So this is what you call metaphase. So they line up over here, and then these centrioles over here, they each attach, the threads attach to the chromosome, to the chromosomes that aligned, and then in anaphase, it pulls them apart. So these threads kind of like retract. They retract back, and when they do that, they pull the chromosomes apart, right? So in here, you can see that now what is going to become cell 1 and cell 2, each one will have an equal number of chromosomes, which means an equal number of DNA. So in anaphase, they're pulled apart, right? And they move towards opposite ends of the cell. It's still one cell, but they move towards opposite ends. So then when they move towards opposite ends, that helps the cell get the signal that now it's time to divide everything, not just the DNA. So now it starts dividing everything into two, and it starts kind of separating, you can see. So during telophase, the chromosomes at the poles, they kind of start separating. this envelope starts going around them, just like we saw in the beginning of how the cell looked like, right? So they separate, the envelopes kind of cover them up, and that's a sign for the cell to start dividing everything else. So we first divide and separate the DNA, and then we separate everything else to make two cells. And these help move around things. So they moved around the DNA, and they stay at opposite directions to let the cell know that it's still dividing. So the envelope, starts coming up around the DNA, and then the DNA uncoils. So it goes from this shape to this shape, because now it's a completely different cell. So now it doesn't need to be in this compact form. It can, like, you can think of it, it can unpack its DNA, and now it's back to this shape. And the cell, cytokinesis, this is when you kind of get rid of this last connection and it becomes two completely separate cells. So we call these the daughter cells. So the daughter cells are, that is the product of mitosis. It's two exactly similar cells with exactly similar DNA. That this is the mother cell, so this is the cell we started with, and these are the cells that we end up with. They're exactly the same as that cell, except now there's two of them instead of just one. So that is the very, very basic process of cell division called mitosis. There's another form of cell division, but we'll discuss that later. I just have a simple video to show it in real time. This is how it really, really looks like under a microscope. These are real cells. These are the threads that are pulling the chromosome apart. See, this is the chromosome. We pull it apart. These are the threads. This is the chromosome. And then we pull it apart. Alright, so that is how it happens. It's really cool in real life. Next lesson, we will be discussing how a virus invades the cells, which is now a really interesting topic to talk about. We're going to talk about how viruses invade cells, what kind of invasions happen, and the effects that has on the body. Or on the person, what kind of effects you see based on what kind of viral invasion that you get. And that is it for today's lesson. I want to keep it simple and short because this is kind of a very, very important topic that you'll see a lot throughout not just high school, middle school, but also in college. So it's really important to understand that first before anything else. And thank you for today's lesson.